interesting work in this group is to demonstrate the graph transmission of a silencing signal. So we use for that what we call the Gus reporter gene. The Gus reporter gene is by a biochemical reaction is indicating the presence of the gene by the blue reaction. So you have here the scion, and here the rootstock is in vitro, of course. So you have in this in this graft you have the scion, which is Gus transgenic. So we introduce this Gus reporter gene in the scion, and here the rootstock, which is non-transgenic. And we demonstrate that when you have the scion, Gus transgenic, but if you have in the rootstock the introduction of a construct which is a Gus silencing transgenic, we have the possibility to silent, to have, you know, to stop the expression of the Gus reporter gene. So this is a technology to have finally a scion which will be non-transgenic, a rootstock which will be transgenic to to stop the expression of undesired genes, as, for example, gene for coding for allergenicity. So maybe someday it will be possible to get this type of selection through transgenic rootstocks. Now, the third and last part of our work inside PILSys is called New Food, and we have tested several varieties across Europe to evaluate how new, better quality fruit cultivars can help to increase food consumption, and we also developed preference maps. So we did that through consumer tests around Europe, in France, the Netherlands, Germany, Poland, Switzerland, Italy, and Spain, with a common methodology with approximately 5,000, which is really important, consumers, 11 varieties of apple, 10 varieties of peach and nectarines. So you have here the place we did the test with the consumers. Some results. 1 minute. Oh, some results. For Apple, the preference map, you know, you have here 68% of the population which prefer sweet and well-balanced acid-sweet varieties. And here, another part of the population which prefer acidic varieties. And you have, for example, for the acidic variety, you have Kanzi, Ariane, and for this group, note Gold Chief, Pink Lady. For the peach, more important, 87% of the population like sweet varieties with this type of variety. Here, 13% prefer acid varieties. So, in fact, we found two major population segments in both products, apple and peach. Sweet and calibrated varieties, which is the most important, acid variety. There is no dependency from age and gender. Certain crop country dependency, but always predominates this metagroup A in peach as in apple, with some difference between countries. Especially here, you have Germany for the sweet variety, which is only 42%. What should we, the chain, be doing? This is a conclusion. The breeders, they have to, with this type of result, they have to do specific breeding for specific population segments. I think it's an interesting conclusion. For the producer, they have to know their markers, take into account flavor beside color and size. For shippers and marketers, separate cultivars not only by color and size, but also by major flavor classes. Communicate also flavor characteristics. For the distribution, they have to cooperate in communication. So, from this group, we conclude that increasing fruit consumption can be achieved when right cultivars are given to the right consumers. Thank you very much. You will see here all the scientists who contributed to this talk and also all the partners involved in Pillar 6. Thank you very much.
We have time for some questions.
Nous avons donc du temps pour des questions.
This is more a scientific question, but I was interested to see your results with the rootstocks and GOS suppression, because we have tried the same with the MELD1, the non-allergenic plants, and tried to put them on rootstocks, traditionally rootstocks, and vice versa, taking roots from these and put conventional apples on them. But we did not get silencing in the other part of the plant. On the grown trees, at least. So maybe it's a difference in the way you're doing it.
Yeah, of course, it's research. You know, it's a research work. It's a fruit is research and experiment. So we have to explore different possibilities. So we demonstrate in the case of the in our case, in the case of the cultivar Gala, that we succeeded, but in a low proportion, is just about 2%. So we have to improve the technology, but we already did the demonstration. So we think that if we improve the technology, and we think that this is possible, we may get interesting results, and why not? Also for allergenicity.